And Will.
All right. Let me see if I can figure this out again. Is that showing up for everybody now?
No.
No. Perfect. I'm a biologist, not a computer person, so bear with me.
You did it well last time.
I'll try that.
There you go.
That's it?
Mm-hmm.
All right. Well, thanks for everybody for joining us. Yeah, this is a pretty important part of our program is outreach and letting the people that we work for know what we're up to. So, yeah, thanks for inviting us, and I guess we'll get started. Yeah, so we'll go through basically the whole program, why we do it, what we're looking for, and I guess some of the results that we've found recently. So, so the sea lamprey is one of the most ancient species on the Earth. It is estimated at 34 million years old. They're native to the Atlantic Ocean. They are anadromous and semiparous, which means that they live in their natural life. They normally live in the ocean, in the salt water, but they return to freshwater to spawn. So they had the ability to deal with freshwater. That's one of our biggest questions is how they can live in the Great Lakes. And then they're very fertile. So each female can carry up to 100,000 eggs. And so then of course they're parasitic on the fish. That's why they're a problem here. So these are a few pictures of some of the damage they can do and different fish species from the Great Lakes that we're concerned about. So yeah, how they got into the Great Lakes. Reports started in Lake Ontario in the late 1800s. Back then they were referred to as Lake Lamprey, and they were found on a lot of the native species back then, like the walleye, lake trout, northern pike. So the thing that saved us back then was Niagara Falls. So lamprey are very determined creatures, but they never could figure out a way to get above the approximately 170-foot Niagara Falls. So that was a natural barrier to them. And then humans started playing with the with the world. So we built the Welland Canal in 1829, and that basically opened up a corridor for them to start their migration. So once that canal was opened up around the falls, it basically opened up their way to spread throughout the Great Lakes. So yeah, by 1839, they had spread throughout the whole lake, or Great Lakes system. By that time, they were found as far as Duluth and Lake Superior. So, and then shortly after that, they were well established. Basically, every suitable river had populations in them at that point. So some of the impacts that they have, we estimate that each juvenile sea lamprey can destroy about 40 pounds of fish, and they do that in about a year and a half that they're in the Great Lakes. So in the 1950s, the lake trout were the native species in the Great Lakes that were targeted for commercial fisheries. So those populations began to crash, and it was a combination of the boom in lamprey populations and overfishing. At that point in time, the lampreys were destroying about 5 times of what the commercial harvest was. So it was estimated at 100 million pounds of fish in the Great Lakes each year that the lamprey were getting. And that basically destroyed the $7 billion economy of the commercial fishing industry. So because the Great Lakes are a unique international ecosystem, so it includes the US and Canada. I believe there's 8 states, several First Nations and tribal areas, and So we needed to come up with a solution for the management from that. So like each state couldn't just manage their own area. So the Great Lakes Fisheries Commission was developed, and that was basically an oversight program to help all those different entities come together for the management purposes. So the Sea Lamprey Control Program was established, and that is a conglomerate of several different entities. So we have the U.S. Fish and Wildlife Service, the Department of Fisheries and Oceans Canada, USGS, and the Army Corps of Engineers. So we all have a unique piece to this puzzle. So the USGS does a lot of the research and chemical research and stuff like that we use. The Department of Fisheries and Oceans and the Fish and Wildlife Service are the agents that actually carry out the control practices. And the Army Corps of Engineers is in charge of a lot of the barriers that we use as blockers in a lot of the bigger systems. So there are several barriers that are built specifically to prevent the migration of sea lamprey. And then we'll talk a little bit about the life cycle. Will, you want to take that section?
Yeah, I could jump in here. Thanks, Nick.
Okay.
And I guess I'll just kind of say next slide if that works for you.
Yep, that works.
So this is kind of our general overview of the life cycle of these animals. We'll kind of dig into each step, and this graphic will be available. It's really handy just to give you an overview. But in general, they'll live about 3 to 10 years total for a grand as a certain stage that we'll get to for a grand total of maybe about 6 to 12 years of life. So these live a little bit longer than we might think initially looking at them. So if we could jump to the next slide, we'll kind of start with their process. So the stage that they first come into the stream at is between March and July. That's a lot of when we do our initial work in surveying with electrofishing equipment and some of the techniques that we'll go over later in the slideshow. But that's when we start seeing these in migrations from adults. And the adults you can see are typically around a foot to a little bit longer than a foot. You might see them in shallower streams congregating. They'll be digging what we call redds in the salmon fisheries. But they'll make these nests that you can actually see them in this slide. They'll grab the rocks with their suckers and build these little horseshoe-shaped mounds. And that allows the water to run over their eggs and keep them oxygenated throughout the season. They'll pair up. Usually there's single pairs. They'll also kind of congregate in masses. But yeah, during this period, they'll come in, they'll spawn, and as we said, they're semiparous, so they'll spawn one time and then die. And the eggs will just kind of hang out once they're fertilized, take a couple of months, and then hatch. And Nick, do you have anything to add on this section of the life?
Nope, that about covers it. They're pretty temperature dependent, I guess. That'd be one thing. So the peak of the run is usually when the water temperatures hit about 14 degrees Celsius. So it can vary widely in our area. Like the southern Lake Michigan, they'll start early March, and then like up in the Keweenaw in your guys' area, we can see them as late as July and sometimes into September on rare occasions. So.
Yeah, awesome. So if we could jump to the next stage. So after a little bit of time where the eggs have been able to gestate in the waters, assuming that the water levels have stayed consistent and they've got appropriate hatching grounds, we'll see the larval stage. So depending on water conditions, so this usually, if there's a lot of nutrients in the area and a good amount of oxygen, this stage will be less long versus in areas with worse conditions, we can actually see this stage extend for up to 10 years. This is the larval stage, so you can kind of see how small they'll start on the far right. These guys are tiny, so when we're looking for them, really just you have to really keep your eye out to spot these guys. But as they get larger and larger, they'll start to transform, which we'll get into in the next stage. But this is the area that we're primarily looking for them at in our surveys, trying to tell if certain treatments have been effective, how long it's been since our last treatment, if we missed a batch or if we were able to successfully treat for the previous year's spawning. But in this stage, you can kind of see, if you look close enough, their eye isn't really fully developed. They don't have a huge amount of coloration. They're largely staying at the bottom of the sediment. You can kind of see in our little graphic image, they'll embed themselves in that sediment and effectively just filter feed. They're not parasites at this phase. They're largely just eating organic material suspended in the water. So they need areas with flow to allow for oxygen, but also things to settle a bit so they can actually get a good amount of food supply. And like I said, based on the conditions of the waterways that they're in, this period can last quite a long time before they'll transform. If they're lucky and they're in an area with lots of nutrients, they can grow very quickly and they can out-migrate as soon as 3 years. But like I said, we've also seen it upwards of 10 and I think upwards of 12 even in certain circumstances, but that's pretty few and far between. And as you can see on our left image here, they reproduce in huge numbers. Single females can produce tens to hundreds of thousands of eggs in one lay before dying. And so we'll see in certain areas, you'll pass over for just a second with our electrofishing equipment and you'll get hundreds of these popping up and just running to the surface or running to the edges of the water. So they're all over the place, and survival, depending on what else is in the water, can be quite high for these guys. Anything else to throw in there, Nick?
Yeah, I guess just to give you guys an idea of how populated they can get. So that picture in the upper left is from one survey site, and we generally cover about 36 square meters. And so all of those lamprey came out about 36 meters. So if you think about that throughout a whole stream system, they can add up pretty quickly.
All right, so the next stage that we see, this is the transforming stage. So this is where they go from what we call larvae or amicetes into what we consider the transforming stage. So this can last between 3 and 10 years in their larval form, and then after a certain point, once they've reached a certain size or they've just gotten enough nutrients to be healthy enough to start that out-migration process, they'll get larger. You can see on the one that's stuck to that person's finger, their eye will develop. It gets much larger and gains a lot more functionality. They'll start to get this silver color. You can see that will get much more developed as they start to head out into the open lakes. We see that with salmon too. There's a silvering that comes with different availability of nutrients. And for the parasitic varieties, we'll start to see much more of a development of that suction plate. You'll see the teeth start to come in. The sea lamprey that we're looking at are parasitic. There are several different species within Michigan that are native. A couple of them are parasites. Several are actually, and correct me if I'm wrong on this, Nick, but there are a couple species that will stay entirely filter feeding throughout their lives. They don't go into that parasitic phase. And the best way to tell, and you might not want to look them in the mouth necessarily, but that's going to be the best way to tell what kind of lamprey or what they'll eat. If they have that really developed bite plate with those rasping teeth, that's generally a good sign that they're going to be a parasite. Smaller mouths are generally just going to be for holding to surfaces and intaking some of that filter food. But at this stage, which is June until March, again, depends on temperatures, depends on water flow rates. All of this can vary quite a bit depending on how the water chemistry is. They'll gradually make their way out to the lakes, generally by the end of the spring. And then lastly, the phase that really we're primarily concerned about is the parasitic juvenile phase. So that's where they're actually out in the lakes. They're attaching to these larger fish, doing damage to our fisheries stock and just life in general, making things really hard. This is kind of where they'll spend upwards of 6 months out in the open water, and within that 6 months, each fish can take in about 40 pounds of live fish. So they're doing a ton of damage for how small they are. And if you think back to how how much they can survive, like how much of these lamprey we see making it into the open lake, that is a huge amount of fish loss per spawning. You know, if one female can lay even at a minimum 10,000 eggs, if most of those survive to maturity and take in 40 pounds of fish when they make it to the open waters, that could be a pretty huge loss of life. Oftentimes you'll see, we get reports from fishermen all the time. They might not even lead to the immediate death of a fish. You'll see these marks like you can see on this right-hand example. You'll see bite marks all over fish from the Great Lakes sometimes, and they'll survive. They don't always kill, but oftentimes, especially when you see these larger fish that have multiple individuals attached to them. Nik showed us a great example this last year of a sturgeon that I think had over 10, right? Something along those lines. It had quite a few lamprey attached to it, and it was still alive. It was not doing well, but it oftentimes you'll see multiple bite marks or even multiple individuals on these fish. And this is where they're gonna get their maximum amount of growth. Lakes are a huge source of nutrients. These fish give them a ton of fat and protein and nutrients that they need to be spawning healthily. And so they'll just eat and grow quite large. And then come that next spawning season, they'll come back to the streams that they initially spawned in. Much like salmon, if you're familiar with their life cycles, they do return to the streams. Typically, shouldn't say always, there's never really an always. But they'll return to the streams that they came from and go back to spawn and then die. And then the cycle kind of just repeats from there. So the area that we're looking at in terms of our treatment and application is the larval stage. Out in the Great Lakes, it can be really hard to actually track down individuals because they're either free swimming or they're attached to other fish. So it can be really hard to track and bring them back. So the methods that we use are we do control with lampricides. So we use one that's called TFM, or I'll try my best not to butcher this, 3-trifluoromethyl-3-nitrophenol. So it's a chemical that's been in use for, I believe, around 50 years now. It's been thoroughly tested as a lampricide-specific targeting agent. It comes in these yellow pellets, so it's really easy to see. But our general process is we start with electrofishing, which is this technique on the right or on the left. We'll go into that a little bit more in depth, but that's how we locate and determine the presence of these animals in their larval stage. We'll walk up and down streams that we believe have been threatened or have historically had populations in the past. And as we walk along, we'll see what individuals come up. Using mathematical models, we can pinpoint which areas need treatment at which points in the year, and then we can then proceed to apply TFM to that area. So another method of detection on the right that we see here, this is actually waterway application of TFM. So we'll load it into what is effectively a leaf blower that is specifically designated for chemical distribution, and we'll apply some to small areas of the lake or stream. And again, it'll cause them to come up to the surface. And that way we can determine how many are in the area and how many have survived the specific treatment of a waterway. So these are just kind of our detection methods that we use to see where the larvae are at and then treat those waterways that they're present in. Nik, anything else to throw in there?
That covers it. Good job.
All right, so just to get a little bit more into backpack electrofishing, some of you who have worked with fish and wildlife in the past or maybe seen just some of the techniques that are used in determining fish presence. Electrofishing is used across the board throughout lots of different scientific agencies in detecting fish. It's effectively, you can see on their back, they have a large battery that has control that allows you to set the different wavelength and frequency of electricity that you're using. So that we can specifically target these lamprey. You can fiddle with those, and you can actually target a wide variety of species with these things. Which river is in the western UP? Oh, that's a great question, Joan. We'll get through that in just a little bit. We have a whole diagram of which ones we're treating. But if we don't answer that towards the end, I'll jump right back on that if that's okay. So when we're backpack electrofishing, we have effectively a positive and a negative, so cathode and a diode. We hold that into the water. This produces a small range electrical frequency that startles and alarms the lamprey. It doesn't really affect many other fish in the area. Some of them, such as juvenile trout, are a little more sensitive to it, and they'll generally just leave. But lamprey, the frequency that we're using causes them to surface out of the sediment. You can't see them free swimming otherwise. They're generally embedded in the ground. Once they come out, we can switch that electrofisher to a slightly higher pulse, and that will stun them. We grab them in, can't really see it, but the ends of these poles have little baskets on them. We'll put them into the buckets that you can see this person on the left carrying. And then once our survey is complete, we'll bring it back to shore. We'll ID what lamprey that we have found. For instance, we'll often see native lamprey mixed in with the invasive. The native lamprey will go back into the stream, and then we'll either collect or just remove the invasives. But we collect data such as, do lampreys have natural enemies? Ooh, that's a great question. So I can answer that real quick. So in general, no. So sea lamprey are not fed upon. Lamprey in general, because of that life cycle, they're not free swimming. They have a very good chance of survival because there isn't much out here eating them. Even on the West Coast, we don't really see a lot of animals eating lamprey. Bears will sometimes. They're very, very nutritiously dense when they come back as adults, but in general, there isn't a whole lot out there that eats them. And that's a big part of what makes them such a danger to the area as an invasive, is there isn't really much outside of us that's removing them from the area. So that's a great question. Let's see. So on electrofishing, in general, it's a really good way to just detect what's in the area without harming the local wildlife. It doesn't impact anything really aside from what it's targeting. You can see certain animals are more sensitive to it, but generally they'll just leave. It doesn't shock them or anything like that. And it just allows us to get a really good look at what's in the area by just on foot going up these waterways and just poking around, seeing what's in there. So in deeper waters, so there's areas that we can't survey by foot. Electrofishing becomes a bit more difficult. You can't really get your paddles in with enough range to see what's going to be in a deeper, wider waterway. So what we'll do is we'll take out these small boats and we'll apply TFM directly, or granular bacillicide, which is another form of a lampreciding agent. And like I said, we'll put them in these backpack sprayers and we'll pick a few small plots selectively based on just kind of an algorithm that allows us to pick based on this size of waterway. Here's how many plots we need. Here's how much of this GB or granular bacillicide we want to apply. And here's just kind of the small amount of area that we want to survey. We don't want to completely flood the waterway. We don't want to overdo a survey, especially in some more sensitive areas. But given the amount that is safe to apply, we'll generally apply it. And then over the course of an hour, we wait to see what comes up from the bottom. And generally, lamprey will respond within about 10 to 15 minutes. It takes a little bit for it to settle. They'll come up, and then we'll catch them, and then same process. Based off of that catch, we can infer how many are in an area. And then after about that hour to, I think, 3 hours, that GB will denature and basically just becomes an inert object. It's coating sand, so you can see on the left-hand side that yellow spray. The yellow is a dye that's applied to the chemical, but then it's just sand that is meant to sink to the bottom, and then after a few hours even, it denatures and becomes completely inert in the water. And here you can just kind of see that monitoring process or the assessment of our data. So we'll often use a lot of our streams that we have to access will go through landowner or Forest Service land. So we need to use oftentimes four-wheel vehicles or ATVs. We'll drive our equipment in, and then after our surveys, those buckets that you saw, we'll empty them into these trays, and that's what allows us to go through one at a time, which, especially when you have a tray that looks like this, can take quite a while because we individually we identify every individual. We measure them, we record that data so that we know in a given area, are we seeing something that could be several years old? Are we seeing what we call young of year or YOI? which are effectively brand new lamprey that are just from the last spawn cycle. And all of this data lets us know, okay, what's our survivorship? How effective has our most recent treatment been? Did something go wrong with the treatment? Maybe a beaver dam was upstream that captured that chemical and didn't allow it to go farther downstream. Things like that let us know how effective we can be in a treatment and if we need to alter our strategy, because this is a really, really long-term project. One thing to drive home is this has been over the course of 50 years and it's still ongoing. So it's gone a long way towards making a huge impact, and it's still an ongoing process. I think we'll jump back to Nik for the stream treatment. Joan, this is kind of aiming towards your question of a bit of where our areas that we target are.
Yeah, thanks, Will. Yeah, so this map just shows a lot of the streams in our area that we cover. So these ones are all positive streams for presence of sea lamprey. This hasn't been updated since last season, so there's a few more that we found up along the North Shore last year. And again, this is just in our jurisdiction. I guess we did forget to mention that. So there's 3 offices that complete the sea lamprey control for all of the Great Lakes. So we have ours here in Marquette, and we have a sister station down in Ludington, Michigan, and then there's one that's based out of Sioux, Ontario, that does the Canadian side. So, and then this map, this shows all the streams that we treated in the 2023 field season. So it's a pretty daunting task for us. I mean, this is, like I said, this is just a subset of all the streams that are positive, but we do a ranking system to figure out how many we can effectively treat in one season. And then those are divided up between the 3 stations. Okay, so this is an example of how much work goes into setting up a treatment. This particular one is the Sturgeon River down in Delta County. So starting at the left, so that river system has a total of 162 assessment sites that we've sampled over the years. So those are options for us to look at and determine how far upstream in the populations of the sea lamprey. So in 2021, we selected 73 of those sites to look at. Those were all checked by staff with the electrofishers. And that took us approximately 22 staff days to complete that work. And like I said, that's just one system, and this is, I would say, the Sturgeon is that like an average size river system for us. It's by no means the biggest, but. And then the far right slide shows after the fact. So that's basically the treatment setup. So all those green dots are where chemical application would take place where the stream is positive. And then the purple dots are on negative tributaries that didn't have sea lamprey in them, but because the water all flows downhill and joins up into the mainstream, we have to account for that water. Otherwise, the chemical dilutes out and it's not lethal to the sea lamprey. So there's a lot that goes into it. So yeah, it just gives you an idea of how we set up a specific stream. So we'll talk about some of the control practices here. These are just some pretty cool pictures. A lot of them are the old school when we first started back in the 50s and up to current time. So yeah, we treat approximately 120 streams every year between the 3 offices. That varies depending on the size and which ones get selected for a year. So our annual budget is usually around $16 million, and that is used to protect the $7 billion Great Lakes fisheries. So the lamprey control team, when they get to a site, they spend the first several days getting preliminary data. It's very important to do all this beforehand to protect the other fish and the non-targets that we're not trying to harm. So the first few days we'll do water discharge measurements and water chemistries, and if it's a new system, we'll do dye studies to get an idea of how fast the river is flowing. But the biggest thing is the gauging that measures exactly how much water is flowing through the stream, so we can meter in the appropriate amount of chemicals. And then the water chemistry actually determines how high of a concentration we need, and it's based off from alkalinity and the pH of the water. So we're talking, like, I would say on average, we're putting in probably like 6 parts per million would be an average one. So it's pretty specific rate. And again, that's used to protect the non-target and the fish that we're trying to protect. So yeah, back in the 50s, there was a lot of research that went into this chemical. A lot of people ask us how we can dump all this chemical in the water and not kill the other fish. But so because lamprey are so primitive, they don't have the ability to metabolize the chemical, and it builds up in their system, and it basically prevents them from uptake of oxygen. So most other fish are able to metabolize it and pass it through their gills at a pretty good rate. So yeah, like I want to say there was 200 chemicals that were tested in a laboratory beforehand before it got approved for use in the stream. So there was a lot of work on the front end of this project. So yeah, once the preliminary data is collected, we start the application process. Like I said, we start at the upper point of the stream and work our way down and try to build up that concentration throughout the system. So obviously in a river, you gain water as you go downstream. So each one of those tributaries will have a feeder set up like the left picture there. And then we will monitor that throughout the shift. So we run a 12-hour chemical bank. That allows us two things. It allows it to build up fully in the water system, and it also allows us to keep the concentration lower. That also aids in helping the fish species. So about every half hour during that 12-hour shift, we will take a water sample and analyze it with a photospectrometer to find the concentration that we're putting in there. And we'll also check the chemistries throughout the day and make our adjustments accordingly. So yeah, a lot of it is done right in the field. We have little portable units that we can do all that analysis with, and we also have on some of the bigger systems, we have a trailer that's set up as a mobile lab unit, so we can run the samples in there for the bigger systems. So another big part of our program is the barriers. Those are used to block the upstream migration of spawning lamprey. A lot of them were purpose-built specifically for lampreys, and some of them are incorporated into hydropower dams, and those are also used as trapping locations generally. So yeah, the barriers, they're kind of controversial now because some of them block migration of like spawning fish like salmon and steelhead and stuff, but in the long run, it helps them by preventing that many more sea lamprey from spawning. So these barriers do block off several hundred to 3 miles to the infestation of lamprey. So a lot of them also have fish ladders built into them that allow passage of this migratory fish species too. So yeah, we kind of work together to try to do the best for everything. And then recently we started looking more at supplemental control methods, and this is kind of where the future of our program is going. So there's been a lot of research into pheromones, which is basically a chemical cue for the sea lamprey of how they determine what streams to come up into to spawn. So the thought is with those, we could put those into streams that are easier for us to treat and attract them into those streams. And then it also helps with our trapping efficiency. There's been some other electrical guidance. So that is the bottom right picture there. There's this array of electrical currents that direct the spawning lamprey into our traps. And in the past, we've done a sterile male release technique. So that was focused on the St. Mary's River. For those of you familiar with that one, it's a massive river. It's way too big for us to treat with our normal practices. So we would sterilize male sea lampreys and release those into the system, and they would spawn with the healthy females. So instead of just taking out that one lamprey, we were effectively killing all the eggs from that female. It was postponed for a while because we couldn't really prove its efficiency in the field, just because it's such a huge system. But it has been brought back in the last couple of years. They're doing another 10-year study down on the Sheboygan River in Lower Michigan. So there's actually a lock and dam system on that river, and the lamprey have gotten above that, and they're actually landlocked in some of the lakes down there, like Burt Lake and Mullet Lake. So it's a much smaller system and closed system, so we can monitor the success of that program a little bit better. And then we're constantly evolving our trap methods for more efficiency to catch more. And then we also trap the juveniles as they outmigrate into the lake. So our hope is to any of the lamprey we miss with the treatment, we can trap them before they get to the lake. So this graphic just shows kind of the timeline of our whole program. The important things to take away here: the peak of the sea lamprey in about the 60s. And you can see the lake trout abundance in the blue colored lines there. Those crashed and were basically non-existent in most of the lakes. So Lake Superior was the only Great Lake to maintain wild populations of lake trout. And then the red line is the lamprey population. So there has been some peaks and valleys for us, but overall we've made a huge dent, and there's actually a wild reproduction of lake trout again. We actually documented wild reproduction in Lake Huron 2 years ago, so that was the first time in a long time. So that was very good to see for us. Yeah, this graphic just shows approximately how many fish are being lost to the sea lamprey. Again, it's pretty staggering if you think about it. And then that bottom right is basically one day's catch out of one of our bigger traps. I believe that one was from the Sheboygan River. So for each one of the Great Lakes, we set a target level that is basically the level of sea lamprey that is acceptable, that will still allow wild populations of lake trout to reproduce. And that's basically our report card, more or less. So each lake is monitored for that abundance. So currently Lake Superior is a little bit higher than we wanted to. Part of that is due to the COVID years. We were not able to travel. So some of the streams had a couple extra spawns that got into the lake that we weren't able to take care of. So Lake Superior did make a little jump on us, but and then Lake Ontario is slightly above currently, but the other three lakes are at or below that target level. So that's a very good thing for us.
Are lampreys edible?
That's a great question. Where they came from, they're actually considered a delicacy, and we've actually shipped lamprey out to the Queen of England before. They make a lamprey pie that is part of their celebration for their country, and they're actually protected over there where they came from now. So, but they do bioaccumulate chemicals from the lakes, so they're pretty toxic, and there's a few of us that have been brave enough to try them, and they're not very good. So I don't recommend it. So again, the whole reason why we're doing this is to support the natural fishery. Obviously, it's a very important thing to our region, between the commercial fishing and the tourist industry and subsistence fishing. So yeah, the fishery in the Great Lakes supports about 75,000 jobs, and it's estimated between $5 and $7 billion annually that it brings to the income. So pretty important. And then obviously, with being such a widespread program, we have a lot of challenges as far as maintaining our social license. A lot of people panic that we're dumping chemicals into the water. and some of the dams are very controversial because a lot of the streams that those are on are notorious for having very good salmon runs. So we've been working pretty hard to bring those ideas together and get fish passage in there and kind of maintain our program with also helping the other species. Yeah, just looking ahead for our program, we are working on a few techniques to improve our methods as far as surveying and taking care of them. So like I mentioned subcon before, that's just alternative methods to control them. We're also doing a lot of research on eDNA right now, which would in the long run would save us a lot of time and effort in our surveys. So instead of us being out there manually looking for the lamprey, we can just take a water sample and filter it, and then it's analyzed in a lab, and that can determine presence of sea lamprey, and it's very very sensitive. So like low density areas where we have a hard time actually finding them, that the eDNA is a lot more efficient at finding those lower populations for us. And I think that's about it. So if we can go on to some questions.
We'll stop the recording now and then go on to